Good evening, students. Today, tips for study. So you must identify which chapter is easy, which one is moderate, and which one is difficult to you. Because some chapters will be easy to some students, some will be difficult for the some students. Similarly, some chapters are moderate to the few students. So you must identify which one is easy, which one is moderate, and which one is difficult to you. It will be vary, depends upon your understanding skills. Clear? For example, the first 10 years I am taking a Neet classes. Among that interaction of so many students. So at that time, few students are said that the cell biology, biomolecules, reproduction in plants, and etc. Which chapters are a moderate one? Which chapters are a moderate one? They said maximum chapters are very easy. For example, living world, mineral nutrition, plant growth and regulators, mostly the plant physiology. So like that, there's so many things are very easy. And the difficult chapters are animal tissues, animal kingdom, biotechnology, molecular inheritance, and genetic inheritance, and etc. So those chapters are difficult chapters. So what's the use of that one, sir? Now I identified so many things are easy, so many chapters are difficult. After that, what we do, sir? So you must identify it. Identify it. Take after that one. Take more time to study the difficult chapters to understand the concept. After that, as usual, you should prepare the MCQs. Clear? So the easiest chapter also. If you feel not interest to study, means at that time you take a easiest chapter, you can study. It will make study interest. So after that study the difficult chapters and revise the MCQs. Clear? So the frequent revision supports to improve your score. Okay, today I will start my session with syndromes. Yesterday we discussed about genetic diseases. In genetic diseases, I gave some examples of aneuploidy, aneuploidy and euploidy. So I will continue that one. So today we will discuss about genetic diseases, specifically chromosomal inheritance. The Klinefelter's which is a genetic disorders, chromosomal disease, which is comes under the non-junction one. The Klinefelter syndromes are comes under the non-junction one, which is caused by Klinefelter syndromes is caused by the additional X chromosome in human. Additional X chromosome in human. The sexual abnormality in male. It is important on sexual in male. Klinefelter syndromes are discovered by Harry in the year of 1942. In the year of 1942, that Klinefelter syndromes are caused by the chromosomal aberrations. Specifically aneuploidy. In Klinefelter syndromes, one chromosomes will be added. So at that time, 2n plus 1. For example, in human being, 46 plus 1. 46, 1 will be added. So automatically that chromosome numbers become a 47. So the chromosomal makeup is a 22AA plus XX. That is a normal. In Klinefelter syndromes, 22AA plus a XY. 22AA plus a XY. So that Klinefelter syndromes are caused by the non-disjunction of XX chromosome. What is the meaning of non-disjunction? So non-disjunction means nothing. The failure of separation of homologous chromosome. Failure of separation of homologous chromosome during cell division. That non-disjunction is discovered by the bridges. Discovered by the bridges. That non-disjunction will be occurs in human being, Drosophila and Datura plant. Datura plant. Mostly non-disjunction occurs meiosis or mitosis or cleavage. Or cleavage. Non-disjunction occurs in mitosis, which is known as a mitotic non-disjunction. Similarly, if it is occurs in meiosis, which is called as a meiotic non-disjunction. Meiotic non-disjunction. So disjunction means failure of separation of homologous chromosome during the cell division. During the cell division. Here in Klinefelter syndromes are caused by the non-disjunction of XX chromosomes. When the abnormal egg with the XX chromosome is fertilized, the sperm with Y chromosome, the resulting the baby consists of a XXY. XXY. For example, the parent mother having the normal 22AA plus XX, the father having the 22AA plus XY. The primary non-disjunction is 22 XX, 22 XX, and here 22A A plus 0, 22A plus X in father, that separation occurs normally. But in the case of female one, non-disjunction will be occurs. Non-disjunction will be occurs. So finally it forms a 22AA plus a XSY. So the son having the Klinefelter syndrome. The son having the Klinefelter syndrome. So what will happen in Klinefelter syndrome? They are the Klinefelter syndromes affected men is a sterile males. Sterile males. The testes are a small one. Testes are a small one. So there is no spermatogenesis. So there is no spermatogenesis. The male sex glands are poorly developed. Poorly developed. The testes, spermatogenesis, those all characters are the sexual one. Clear. So that male sex glands are poorly due to chromosome. Hence, so smaller. So there is no spermatogenesis. Clear. So that's why we are saying that Klinefelter's affected male is a sterile males. It will be affects the male, sterile male. That is important one. Klinefelter's affect the sterile male. It having the 47 chromosomes, 22AA plus a XXY. That is most important one, ma. Similarly, the Klinefelter affected Klinefelter syndrome affected male, they have a breasts are enlarged and tall. The male hormone quantity also low. The male hormone quantity also low. Hence, they are mentally affected. Hence, they are mentally affected. Those are the important characters of a Klinefelter syndrome. Clear? The next one is a Turner syndrome. genetic disease in human filters is affected in Turner syndrome is affected in a female which by the absence of 1X. In Klinefelter syndromes, excess of one chromosome. Here, Turner syndrome, absence of 1X chromosomes. Absence of 1X chromosomes. So Turner syndromes are sexual abnormality in female. Sexual abnormality in female, that is the most important one, which is discovered by Turner in the year of 1938. The Turner syndromes is discovered by the Turner in the year of 1938. That's why the disease syndromes are called as a Turner's syndrome. The Turner syndromes also caused by the chromosomal aberrations of aneuploidy. Chromosomal aberrations of aneuploidy. Aneuploidy means, you know very well, deletion 2n-1 will be lost. This is called as a Turner syndrome. So here Turner syndromes, what will happen means 22AA plus XO will be there. 22AA. 22AA plus X will be there. 45 chromosomes instead of 46 chromosome. Abnormality is due to the 45 chromosome instead of a 46. The missing of one chromosome is a 1X chromosome. The missing chromosome is a 1X chromosome. Hence the chromosomal makeup is a 22AA plus XO equal to 45 chromosomes. 45 chromosomes. That Turner syndromes also caused by the non-disjunction of XX chromosome. Kindly note it. The Turner syndromes caused by the non-disjunction of XX chromosome. When the abnormal egg without any chromosomes, when the abnormal egg without any X chromosome is fertilized by the sperm with X chromosome, fertilized by the sperm with X chromosome, what will happen? The result is a XO. Result is a XO because the abnormal egg without X, that thing only fertilized with the sperm having the X chromosome. So automatically that result is a XO. O means a absence, nil. Clear? Which the baby consists of a XO chromosome. So the baby develops into the sterile male. Sterile male. Just now I told Turners are just opposite to the Klinefelter syndromes. There male, here female. So the Turner syndromes affected female is a sterile female. The female phenotypes, she has a female phenotypes, but there is no menstruation. If the particular woman affected a Turner syndrome, she have a female phenotype. She have a female phenotype, but there is no menstruation. Clear? So these are represented by the ridge of white tissue, which is called streak gonad. Ovaries are represented by the ridge of whitish tissue is called streak gonad. Here you can see the diagram. The parental mother is a 22AA plus XX, mother is a 22AA plus Y. So you know very well that normally which is undergoes a segregation process in mother 22A plus XX and 22A plus O. Here the disjunction is occurs, that XX chromosome failures to the separation. In father, as usual, normal process, the gametes will be segregated as usual. 22A plus X, 22A plus Y. You know very well, A is a autosome, XY is a allosome. So that during that gamete formation, that allosomes will be segregated into a haploidic one. After that fertilization, during that fertilization, the gametes will be formed. That is a normal protocol, you know very well. So that gametes, the segregated gametes will be on the undercourse of fertilization, fine. At that time, you can get it 22 AA plus A plus XX. 22 AA plus XO. Clear? 22 AA plus XO. Clear? Again, I repeat, ma. So, Turner syndromes, mother having the 22 AA plus XX, father having the 22 AA plus XY, that is heterogenic one. That gamete will be segregated 22 AA plus XX and 22 AA plus the XO. Here disjunction is occurred. So in father, normally segregated 22 A plus X, 22 A plus Y. So after that, which is undergoes a fertilization process, as a result, 22 AA plus a XO, which is called Turner's syndrome. She is a Turner syndrome. Fertile female. Next one is a Turner syndrome. Sorry, Turner syndrome just now we completed. Next one is a Down syndrome. Down's syndrome. The Down's syndrome is also known as a Mongolism. This is an important one. The Down syndrome is also known as Mongolism. The boy affected by the Down syndrome, Mongolism, also known as a Mongoloid. Also known as a Mongoloid. The Down syndromes are a Down syndrome abnormality was very first discovered by the Down in the year of 1866. That's why that syndrome is also Down only identified the abnormality. That's why the disease is also known as a Down's syndrome. Clear? That Down syndrome is also caused by the chromosomal aberrations due to the trisomy. Yep, trisomy of autosome. Trisomy. Here, that 47 chromosome instead of 46, that chromosome number 21 is represented by the 3 copies. Hence, it is caused by the autosomal aneuploidy. Caused by the autosomal aneuploidy. It is that the Down's syndrome also arises by the non-disjunction. So that the 3 chromosomal diseases are raised by the non-disjunction chromosomes of 21 pairs during meiosis. The Down syndrome occurs by the action of non-disjunction of chromosomes of pair of 21 during meiosis. Here, hence the both autosomes of this pair enters the same egg. Autosomes of this pair enters in the same egg when the egg is fertilized by the normal sperm. So the Down syndrome results are that nose is oblique, the ears are, you can see the picture, ma. The nose is oblique, the ears also malformed, not in proper manner. You can see the picture. Ears are malformed. They are mentally retarded. The neck is also short and broad. The mouth is also, you can see the picture. The mouth is also constantly open. The mouth is also constantly open. The syndrome, the Down syndromes appears much more frequently in children's born to women in the lateral part of reproductive life. The Down syndrome appears much more frequently in children born to women in the lateral part of reproductive life. Clear? So just you see the diagram, the mother having the 22AA autosomes plus XX, the father is also having the 22AA plus XY, the normal chromosomes. When which is undergoes a segregation process in cell division process, at that time the 22A plus A plus X, 21 A plus X. So here also the disjunction is occurs in the female one. That man normally it will be segregated 22A plus one allosome X, similarly 22A plus Y. So while the gamete will be fused during the fertilization process, at that time we can get it at 22A plus A plus XS, because that is called as a trisomy. This is called as a trisomy. The trisomy causes the Down syndrome. Clear? So now I explained already that what are the characters of Down syndromes. Mongolism. The nose is oblique, ears are not malformed, ears are malformed, the mouth is constantly open, the mentally retarded. So this is called as a, the neck is also short and broad. Those are the important characters of a Down syndrome. The next one is a Mendelian disorders. Next one is a Mendelian disorders. Mendelian disorders. The Mendelian disorders are chiefly determined by the alteration or mutation in single gene. So Mendelian disorder, Mendelian, why which is called as a Mendelian disorders means Mendelian disorders is also known as a Mendel disorders. So the genes are responsible for all. This statement is strongly believed by the Mendel. So gene is important on in Mendel's experiments and etc. etc. So here that the alteration of mutation in a single gene, which causes so many disorders. That diseases or disorders are called as a Mendelian disorder. That disorders are called as a Mendelian disorders. Many disorders is there. For example, phenylketonuria, albinism, thalassemia, hemophilia, sickle cell anemia. So many things will be there. Which is also known as a metabolic diseases. Some metabolic diseases, for example, phenylketonuria, alkaptonuria, albinism. Those are the comes under the inborn errors disease. Those are comes under the inborn error disease, which is also known as a metabolic diseases. We will discuss one by one. The first genetic disorder is hemophilia. So hemophilia, hem means a blood related one. Hem means blood related one. Hemophilia means the lack of clotting factor in lack of clotting of blood, which is caused by the single protein helps to the blood clot, which is affected. So the hemophilia caused by the single protein which helps in blood clot is affected. So hemophilia is caused by the sex-linked recessive disease. The hemophilia is a sex-linked recessive disease, which is inherited disorder of blood. You know very well, all genetic diseases are inherited. Similarly, the genetic diseases are, we cannot cure it, we can control it, we cannot rectify it, we can control it. Keep it in your mind. The genetic disease, there is no remedy. Genetic disease, we cannot cure it. We can control it. Otherwise, we clear. For example, your father or mother having the diabetic in a 30 ages means if you are maintaining the control diet or etc. means you can postpone that particular disease. Still, the genetic disease we cannot cure it. So that hemophilia inherited disorder of blood in which essential clotting factor either partially or completely missing, because which is that particular factor is important for the blood clotting. That blood clotting factors, either partially or maybe completely absent in blood, because blood clotting is very important, you know very well. While we get an accident, at that time blood clot is important one. So in this way, blood clotting, in this way we can prevent the loss of blood. So that there the blood clotting factor is so important. There is no blood clotting factor means the blood will be undergoes a continuous bleeding process. We will get a blood loss. So automatically it's very dangerous. It leads to the dangerous condition. So in order to avoid means the blood clotting factor is the more important one. So in this way that for the blood clotting factor partially or completely absent means which leads the absent in the bloods, which leads the hemophilia disease. That hemophilia disease also called as a royal disease. Hemophilia disease also called as a royal disease, because the Queen Victoria, very first the Queen Victoria was carrier of disease. So that's why the disease is also known as a royal disease. Kindly note it, ma. The next one is a thalassemia. Next one is a thalassemia. So thalassemia means due to the autosomal mutant gene, due to the thalassemia is caused due to the autosomal mutant genes. Thalassemia caused autosomal mutant genes. Here, defective synthesis of hemoglobin, specifically in the subunits of hemoglobin, alpha and beta globin chain. So the changes occurs in the alpha and beta globin chain of hemoglobin. So thalassemia makes a too anemic, because the disease also that blood related one, because the hemoglobin is more important one, you know very well. Here that subunits of hemoglobin will be affected. So defective hemoglobins will be produced. Hemoglobin means here defective subunit of hemoglobin will be produced. So automatically, who suffered from the thalassemia, automatically that person become anemic. So that affected persons, that oxygen carrier level will be very low. Clear. The next third genetic disease is a sickle cell anemia. Sickle cell anemia. So that autosomal linked recessive disease. The sickle cell anemia is a autosomal linked recessive disease. I think we already discussed in earlier topic of inheritance and variation chapters. So that sickle cell anemia is autosomal linked recessive disease. Sickle cell anemia controlled by the two alleles. What are the means? HPA and HPS. Here HPA is a normal one. So one person having the HPA gene and HPA gene means that person is a normal one, does not affect the sickle cell anemia. That HPS is affected. That both genes, HPA, both genes becomes a HPS. HPS means that person is affected by the sickle cell anemia. If partially changed in the alleles means, for example, HPA and HPS. HPA and HPS means that person is a carrier. That person is a carrier. It is not a exposed. That is called as HPS and HPA and HPS. Clear. So that sickle cell anemia is a blood disease. Kindly note it. It is important. Sickle cell anemia also blood disease. That RBC becomes a sickle shaped. Also, RBC becomes a sickle shaped. Hence, oxygen carrying capacity also reduced. Here you can see. So that is beta chains. That there is a beta chains globulin. That hemoglobin is made, you know very well that hemoglobins are made up of two subunits, alpha chain and beta chains. In this beta chains, hemoglobin of beta chains, that so many amino acids, that code amino acids will be a amino acids codes are present it. So amino acids codes are present it. You know very well that codes are responsible for the production of amino acids. That amino acids can able to make the protein. If once the protein is synthesized, once the protein is produced, means that gene is expressed, because gene will be expressed in the form of protein. Gene will be expressed in the form of protein. So which one is base? So amino acids, that codes is very important, because the codes are responsible for synthesis of a particular amino acids. That amino acids can able to synthesis the proteins. The proteins means once the protein is synthesized, the gene is expressed. Clear? Because gene is expressed in the form of protein. Here, there's so many amino acids will be there. Valine, histidine, leucine, tyrosine, proline, glutamic acid, glutamine, air, etc., etc. You can see 1, 2, 3, 4, 5, 6 in a sixth position. In the sixth position of beta chain, having the GAG codes for the synthesis of glutamic acid. The code is altered. The first line is a normal one. Here the second one. The second one is a valine, histidine, leucine, and proline, etc., etc. Here the codes is changed. GAG is changed into the GUG. Here that A, here normal gene having the A in between the G. So GAG codes are responsible for the glutamic acid synthesis, but the mute that affected one, so that A will be replaced by the U. Here the total changes is occurs in the single base. The codon, that single codes are very important for the synthesis of specific amino acids. So CUG is responsible for the valine synthesis. GUC is responsible for the valine synthesis. So in sixth position of beta chains of hemoglobin, glutamic acid, glutamine will be replaced by the valine. Clear? So this is called as a sickle cell anemia due to the changes in the sixth position of amino acids. Clear? Which leads the sickle cell anemia. This is the most important one. So many times that question is asked in any previous NEET exams. So kindly note it. So glutamic acid is replaced by the valine at the sixth position of beta chains of hemoglobin, which is called as a sickle cell anemia, which is a blood disease. Clear? Next one is Urea. Just now I told so many diseases are comes under the inborn error diseases. Inborn error diseases means there is a genetic disease because that particular diseases, particular character will be inherited from one to next generation. That is called as the inborn errors, which is also known as polyketuria and alkaptouria. Third one is the albinism. The first one is the polyketouria. So polyketouria is a metabolic diseases which is characterized by the continuous accumulation of phenylalanine in the blood. So important one, kindly note it. So the polyketouria means a continuous accumulation of phenylalanine in the blood. Phenylalanine in the blood. Why means the particular enzyme, phenylalanine, hydroxylase is important one. But if that person is affected by the polyketouria means that enzyme is not produced. That phenylalanine hydroxylase enzyme is not produced because that enzyme is responsible for the conversion of phenylalanine into the tyrosine. So you know very well that continuously if you are taking anything, it means that thing should be converted into the another one thing. In this way, that energy will be extracted by the tissues of our cell. The remaining things will be excreted throughout our body. So one matter should be converted into the another one. Then only it should be a useful one. Here in polyketouria, so particular due to for that conversion, so many enzymes will be essential, universal, because enzymes are a catalyst. The enzymes are a biocatalyst. Commonly, universal enzymes are a biocatalyst. Here, that the phenylalanine hydroxylase is important for the conversion of phenylalanine into the tyrosine. Here that the recessive genes are here the phenylalanine hydroxylase is not produced. Hence, that the phenylalanine will be continuously accumulated in the blood. So that excess, if continuously accumulated in the blood, means that excessive phenylalanine is excreted in the urine. The excess of phenylalanine is excreted in the urine. So the children is suffering from this disease are known as phenylpyruvic idiots. Important one ma. The children is suffering from this disease are known as phenylpyruvic idiots. The particular disease is characterized by the mental defect, feeble mindedness, and hypertonicity, and light pigments. Just now I told that inborn error disease, metabolic disorders, we cannot cure it, but we can control it. So this polyketouria is also controlled by the phenylalanine free diet, because if you are taking so many foods, means it should so many proteins are present it, so that particular amino acids also present it. So if you must avoid the phenylalanine free food, because already the excess phenylalanine will be accumulated in your blood. So we must avoid phenylalanine free diet. Clear? So polyketouria, it is a metabolic disease. It is an inborn error disease. It is a hereditary disease. Here, recessive characters caused by the recessive genes. So those that are recessive genes are present. Those recessive genes are present, that enzyme is not produced. So automatically there is no conversion. That phenylalanine will be continuously accumulated. That's all about the particular genetic disease of follicuria. The next one is alkaptouria. 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 So alkaptouria is also metabolic disease characterized by the accumulation of alkapton. Alkapton means a homogenetic acid in the blood. So accumulation of alkapton in the blood, which is called alkaptouria. So alkaptouria is the inborn error metabolism, which is also heritable diseases. The recessive characters caused by the recessive genes represented by the HH. That recessive genes are represented by the HH. That alkaptouria also simple Mendelian characters, which is inherited like a Mendelian monodihybrid cross. So it will be inherited like a Mendelian monodihybrid cross. The recessive genes are present, that enzyme homogenistic acid oxidase is not produced. Homogenistic acid oxidase is important for the conversion of phenylalanine to the tyrosine to the acetoacetic acid. If that enzyme is not produced, if its enzyme is not produced, means there is no conversion. So continuously that alkapton will be accumulated into our blood. Clear? Accumulated into your blood. So when these enzymes are absent, alkapton means a genetic acids is derivatives. The alkapton is a derivatives of tyrosine. That alkapton cannot converted into the acetoacetic acid due to the absence of the enzymes. So which results continuous, that results automatically that alkaptons will be accumulates in the blood. So that urine, which persons affected by the alkaptouria, that person urines turns into the black when exposed to the air. That is also important question mark. Which persons are affected by the alkaptouria? That person's urines are turns black when exposed to the air. So the X is homogenistic acid. Alkaptons are attached to the collagen of cartilage and another connective disease due to the ear and ear. So those are the important things of alkaptouria. The next one is the last albinism. Everybody's albinism means it also hereditary disease. All so melanin pigments are where the melanin pigments are absent from the skin, hair, eye, or etc. So etc. Hence, that affected persons are pale in color. Other eyes are brown in color. Hair, eyeball. So normal person's eyeballs is black in color, but albinism affected person's color is brown in color. Because that melanin pigments are absent. So which is important question mark. Melanin pigments only determine the colors. That means that color is a pale color. So that persons are called, that is also important. Albinois means who suffered from the albinism, which is called albinois. That person is called albinois. That albinois are highly sensitive to the light. Highly sensitive to light. I think you can see the lot of persons, so that fully white in color, include hair and eyeballs also like a white brown color because absence of that pigments. Clear. So that persons cannot tolerate that light, cannot tolerate, right? That eyesight is also too low. Eyesight is also too low. That persons are highly sensitive to the light. The recessive hereditary disease is caused by the recessive genes of AA. That albinism is also caused by that recessive genes are represented by A. The recessive genes are present that enzyme tyrosinase cannot be produced. Hence, the tyrosine cannot convert into the melanin pigment. So normally the tyrosine will be there. You can see the flow chart. Normally the tyrosine is present. So due to the presence of tyrosinase, so carboxylase, nuclease. So nuclease will be act on the nucleus. Lipase will be act on the lipids. Proteinase will be act on the proteins for this conversion. Like that, tyrosinase will be act on the tyrosinase. So and the tyrosinase will be act on the tyrosine. The tyrosine will be converted into the phenyl hydroxy phenyl pyruvic acid, that which is converted into the melanin. In case if tyrosinase is not produced, means there is no conversion of the tyrosine, automatically it will be accumulated in blood. There is no conversion. So finally we cannot get it a melanin pigment. Clear. So which is called as albinism. So that's all about the genetic diseases. So that last easiest is called as a pedigree analysis. Pedigree means the systematic listening of parents and the grandparents of individual. It is a family tree. The pedigree analysis means simply we can say it is a family tree for a large number of individuals. The pedigree analysis is the family tree for a large number of individuals. The pedigree charts contains the different types of symbols. It is the most important one. Surely one question will be asked from the pedigree analysis side. Clear? So they will give on diagram. You must understand, and you should give the answer for that one. Clear? So pedigree charts consist of the different types of symbols to indicate the different types of individuals. For example, that normal square box, that normal male members of family shown the square box and females by the circulars. That normal square box, first one you see the diagram only. That square box indicates the normal male. The circle which indicates the normal female one. So that normal female one. That between the square box and circle one, one line is there. That horizontal line is there. That is called as the mating. The mating represented by the square and circle connected by the horizontal line between the square and the circle. The last one is parents and their children are represented by the connecting of children by mating the line of parents. Here the last one, the identical twins are indicated by the children connected by the inverted V line and horizontal line. So V line with the horizontal line, which is called as identical twins. Here, non-identical twins means options of that horizontal line in between the circle. Non-identical twins are indicated by the children connected by only inverted V line. There is no horizontal line. Clear? Next one, represented by the closed and female that close the square box and round circles are shaded by the black in color. So that square and circle shaded by the black in color means that square and females are the circles are affected males and females. That half is shaded, the square is shaded with the half, remaining is not shaded, which is called as a heterozygous male. Heterozygous males are half closed circles and females means half closed circles, half shaded things means heterozygous. That it is that person is a carrier one. Clear? That carrier of sex-linked recessive is represented by the circle with not inside. Here carrier. The carrier means the carrier of sex-linked recessive is represented by the circle with dot inside. So that round circle means female, you know very well. One small dot that fully shaded means that person is affected, whether maybe male or female. That means he's carrier one. She's a one. The next one is that question marks. Question mark is placed in the center of the circle. Is one question mark is present means that symbol indicates the uncertain phenotype of individuals. Uncertain phenotype of individuals. Similarly, the in square box is present. The question number is present in the center of the square box means death of an individual is represented by the closing up square box. Means which means uncertain male. Uncertain male. The last one, that crossing in the square box. The death of individuals is represented by the crossing the square box or circle. So in cross line, in square box or circle means death of the individuals. For example, the particular population is there means some peoples may be a death due to the age factor or maybe any other things. So which indicates that crossing the square and circle, which indicates the death of individuals once. Clear? So those are the important symbols of those are the important symbols of those are the important symbols. Just just to give an answer. You can give the answer. If any question is asked from the pedigree analysis, you can keep the symbols in your see. The square is the male, circle is a female one. That unspecified means we don't know whether it is a male or a female. That fully shaded means the affected individuals, affected individuals. The line, horizontal line between the square box and circle means mating. Similarly, that two horizontal lines between the square box and circle means that mating bit related. Some for close relatives, you know that the horizontal lines, the horizontal line connected the square box and circle and get the progeny like a horizontal line, square box, which is also interconnected, means that parents above the children's below. The parents, that male mating with the female one, that F1 generation will be produced, that F1 generation also female and male. Clear. So the next one is so that when the mating is here, when this mating is occurs between that normal male, which indicates the normal, which indicates the female one, between that time is a square with shaded, so that progeny is affected by the diseases. The parent with the male and child affected with the disease. Here that there is no disease for male and progeny got the diseases. So that individual's grandfather or grandmother may be a carrier for the particular disease. So in parents' generation, it is a recessive one. So after that, it becomes a dominant one. The last one. So halfly circle or dot means that is called as a carrier. So that crossing line between the square box or circle means a death ratio. Clear. So like that, those are the important symbols. Twin means that V-shaped one. So if which is one horizontal line is connected the male and the female means which is called as identical one. Non-identical means the other horizontal line is absent. That is called as a non-horizontal one in a V-shaped one. Clear. Those are the important things. Also that question. And here mating between the affected male and normal female one. At the one male and normal, one female, one male normal one, and one female and two female and one male is affected by the diseases. Similarly, so it will be you can diagram you understand. So you should most important one. Clear. So that's all about inheritance and variations. That's all about inheritance and variations. So inheritance and variations, what are the things we discussed? So genes and Mendel monohybrid and dihybrid experiments, Mendelian characters and Mendelian's laws, exception of Mendelians and post-Mendelian process, chromosomal theory comes under the cytoplasmic inheritance, comes under the post-Mendelism, and mutation part. And the last one is genetic disorders. Those are the important topics, comes under the principles of inheritance and variations. So many people feel that topic is too tough, not like that. Concept and you can each and everything. So next we will discuss a few one or two questions in inheritance and variations. That questions is also asked in the previous Neet exams. competitive NEET exams like AMT, AMPMT, and AIMS, AFMS. I think you know very well. AMT means All India Medical Test, prelims and mains. AIMS also you know very well. And what's the meaning of AFMC? Anybody knows? AFMC. AFMC means Armed Forces Medical College, abbreviated into AFMC, Armed Forces Medical Colleges, which is leading training institute located in the Pune, Maharashtra, located in the Pune. The Indian Army. That AFMC is managed by the Indian Army forces. So approximately that AFMC may have the 130 seats, I think 130 seats. Among that one, mostly they will give seats for boys. Approximately, I think 105 seats for boys. Remaining seats for girls. Clear. So AFMC means Armed Forces Medical College, managed by the Indian Army Forces. That is important. Indian Army Forces. So you can get a seat in there also. So you would, if you want to sit in AFMC means there also NEET is important, because there is no separate they are not conducting a separate examination to join there. So they are also NEET. You should clear the NEET. Clear the NEET means you should get a high score. So you can get a AFMC also. Clear. So important questions. So many important questions will be there. Previous questions. The first one is two unrelated individuals are lines are often phenomena is called. Clear my two unrelated individuals or lines. Two unrelated individuals or lines are crossed. The performance of F1 hybrid is oftenly superior to the both its parents. This process is called as the phenomena is called as which one? Transformation, splicing, or metamorphosis? Heterosis. Heterosis. Because transformation means it will be transformed from the axis to the specific one. That is called as a transformation. Splicing means you know very well, cutting process that comes under the molecular biology. Splicing, removal of mRNA, introns removal, that is called as a splicing. Metamorphosis means which is comes under that cleavage part or etc. etc. So heterosis is the answer. Clear. Next question. Test cross or in Drosophila involves crossing. The test cross, you know very well what's test cross, the parent recessive one, that F1 generation is crossed with the recessive parents. That is called as a test cross. So the test cross in plants are Drosophila involves the crossing between the two genotype and recessive traits between the two F1 hybrids. The F1 hybrid with the double recessive genotypes between two genotypes with the dominant traits. Which one is correct answer? Which one is correct answer, ma? So I will give explanation. In test cross, F1 individuals showing the dominant phenotype. Showing the dominant phenotype, which is crossed with the homologous recessive parent. So the progenies of such cross can easily analyze to predict the genotype of test organism. I again give the description for that question mark. So in the test cross, F1 generation shows only the dominant phenotype. That is a cross with the homozygous recessive parents. So that progenesis of that progenesis are easily analyzed to predict the genotype of test organism. I think now very easy to say answer because I gave explanation with the double recessive genotype. F1 hybrid with the double recessive genotype. Clear? The next question one by one. Any one we can discuss it. The next one, the fourth one. Which of the following variations are not variations are temporal and having nothing to do with the variation, discontinuous variation, acute variation. The last one is all of those. Hereditary variation means it will be transmitted, inherited. Discontinuous means it is not continuously traveled. Acute means it is received from some sources. That is called as acute. So your answer is acute because acute variations are caused by the environmental factors. I think in introduction of principles and variations, I gave that definition for 2-1. Genotypic variation and one is the phenotypic variations. So in the phenotypic variations, I explained about it one. Clear. So acute variations are caused by the environmental factors such as food, temperature, or other ecological factors. Those variations are temporarily and have the nothing to do with the last or next generation. Clear. So again, I'll give the description. Acute variations are caused by the environmental factors like a food or etc. So those variations are temporary and have nothing to do the last or next generation. Clear. For example, loss of limb in accident, loss of limb in accident. So answer is acute variation. Because acute variation is temporary variations because which is caused by only the environmental factors. So environmental factors. So clear. The phenotypic variations, simply you can keep it in your mind. The phenotypic variations are not inherited, but genotypic variations will be inherited. So answer is acute variations. Answer is acute variation. And the seventh one, any question you can take and the test cross is carried out of. The test cross is carried out of. Your options are determine the genotype of plant at F2, predict whether two traits are linked, assess the number of alleles of genes, determine whether two species or variants will breed successfully. breed successfully. Which one is the correct answer, ma? The test class is carried out. So if you are in MCQs, you must prepare like this. So that's why I'm explaining. So if one chapter is completed means you have to prepare a MCQs in a year long. If so many things we will discuss like this, clear. So we will discuss in discussion part. So you must discuss like this. Sir, you must prepare a question bank like this. After that, you have to discuss it. Clear. So test cross is during the discussion time after preparation of question papers. You should understand the questions. After that, only you should see the options. So many students are first they're seeing that options. So if you see the options, surely it will make a confusion. So you should read the question paper twice. Questions twice. You must understand that questions. After that, you can see the options. So while you are reading the question paper and when you get the clear-cut idea about that questions, so automatically your mind will tell some answers for that one. So after that, if you are reading that options means surely you will get the answer. Clear. So that is seventh one. The test cross is carried out to determine the genotype of plant at F2 because the test cross is performed. The test cross is performed to determine the genotype of F2 plant. Performed to determine the genotype of F2 plant. In typical test cross, an organism showing dominant phenotype and those genotype is to be determined is crossed with one that homozygous recessive for the allele being investigated instead of the self-crossing. Though the progenies of such cross can easily be analyzed to predict the genotype of the test organism. Clear? You can say the 12th question, ma. 12th question. So which one of incorrect statement with regard to the importance of pedigree analysis? Just now it is we discussed which one of the incorrect statement. So the statements are there. Which one is incorrect? Which one is incorrect? Which statement is incorrect? With regard to the importance of pedigree analysis. So regards pedigree analysis, which the following statement is incorrect. That is the question. So your options are it confirms the DNA is the carrier genotypic information. It helps to understand whether the traits questions is dominant or recessive. It confers the traits linked to one of the autosomes. That pedigree analysis helps to the trace of inheritance of a specific traits. Your answer is 12th one. Your answer is A, because pedigree analysis confers that DNA is the carrier of genetic information. Because in just now I told DNA is a carrier, because while that mating process occurs between the normal male and female, so if in case if you get affected, if the progeny is affected by the particular diseases, means that grandfather or grandmother may carrier. That gene is recessive in the parental case. It will be expressed to the grandson or maybe a granddaughter. So that characters, that affected carriers, the disease is affected by disease is caused by the particular genes because of some mutation is there. Clear. So that diseases will be carried from one to another generation. In one generation is a dominant, another generation is a recessive. The next generation it is a dominant one. So that diseases causing gene, that recessive genes will be transmitted through DNA only. So the first question confers the DNA, the genetic information. Clear. The pedigree analysis does not understand the does not support to understand the traits, characters. Clear. Similarly, the pedigree analysis not linked with the another autosomes. Does not pedigree analysis does not helps to trace the inheritance of specific thing. Clear. The next one, the next question. 13th one. Just now we discussed which of the following is true for the recessive disease in family A and family B? Which is the following is true for a recessive disease in family A and family B? In family A, normal male is mating with a normal female one. In family B, affected male normal female. So at that time, family A progeny, one male and one female is affected. One male and one female is affected. But in the case of family B, only one female is a normal female. Remaining three males are affected, one female also affected by the diseases. So which of the following is true for recessive gene in family and A and B? So your answer is, just you find the answer, ma. In family A, both parents are homozygous recessive. In family B, both parents are homozygous dominant. Option C, in family B, both the parents and heterozygous are recessive. Option D, in family A, both parents are heterozygous recessive. Your answer? Correct. In family A, both parents are heterozygous recessive. The option, that last option D is correct. Clear. The family A, both parents are heterozygous recessive is correct. So why we are selecting, why the D answer is correct? Any idea? Because in family A, if both parents are homozygous recessive, what will happen? The both should be diseased and should have the 100% of diseased progeny. But we did not get a 100% of diseased progeny. We are not received. We received two progeny only, affected progeny. Remaining three is a normal one. Clear. So in family B, if both parents are homozygous dominant, they would not have got the recessive disease in first place. If both are heterozygous recessive, then also they would not have got the disease. Neither 80% of progeny will be diseased. Clear? So your answer is a D. So in this way, you can discuss so many questions. Clear? You can think of so many questions. So many things will be there. For example, 20th one. So last only one question we will discuss it. So in his classic experiments on pea plant, Mendel did not use seed shape, flower position, seed color, or pod length. Which one is correct, ma? This is the easiest question. Classic experiment did not use pea plants. As a pea plants, he did not use the following one of the characters. Answer is a pod length. Pod length. Because he used the plant height, flower position, pod color, pod shape, flower shape. So like this, the questions may be asked directly. Questions may be asked directly. Otherwise, question you can understand that. If you understand that concept means you can think and give some answers like a pedigree analysis questions. Clear. Sometimes questions will be asked indirectly. Questions will be asked in a So the last only question, ma. Alleles are different molecular factors, different phenotypes. The 17th question. Types, true breeding, homozygotes. Which one is correct? A. Different molecular forms of genes. I explained alternative of genes. Alternative forms are known as different molecular forms of genes. Clear? It is not a heterozygous. It is not a heterozygotes. Clear? So alleles are a different molecular forms of a gene. That is a correct one. The last 8 question also. 18th. Multiple alleles are present in where? AM 50 2015 means conducted and cancelled with some reason. So multiple alleles are present in where? The first option is correct. The same locus of chromosomes. Chromosomes. Because so many times we discussed that locus. Locus means a location. So at the same that multiple alleles are present in the same locus of chromosomes, but it controls the single character. So multiple alleles are present in the same locus of chromosome, but it controls the single character. That is the major. So multiple alleles. That is not an ancestor one. It is not present in the different chromosomes. Clear. It is not present in a different locus on the same chromosome. It will be the multiple alleles are present on the same locus of the chromosomes. Clear. I think like this you can discuss questions. You have to prepare questions and discuss on. So pleiotrophic gene 21 ton. The pleiotrophic gene. Pleiotrophic genes controls the traits only in combination with another gene. Pleiotrophic genes controls multiple traits in individuals. The pleiotrophic gene expressed only the primitive plants. Pleiotrophic genes involved during Pleiocene. Which one is correct? Which one is correct, ma? Your answer is a B. Pleiotrophic genes controls multiple traits in individuals. Multiple traits in individuals. So now we discuss over few questions. Few questions. In these sections, in this way, you can prepare a multiple MCQ questions and just you reading the past Neet questions. So just you can easy to find some things. So just now I told in pedigree analysis side, surely they will ask one question. Similarly, that gene interaction side, they will ask one question. Gene interaction side, they are asking on questions. And Mendel's experimental side, most probably in the character side, character selection side, they are asking on questions. Clear? And very rarely they are asking that questions from the mutation side also. So surely that gene interaction and Mendel experimental side and pedigree analysis. So surely they will ask 3 questions from the particular areas. Clear? So we have to concentrate much more. We have to concentrate more than the other areas. Clear. So in this way, you have to prepare a question paper and analysis and put some analysis. Clear. So then only easy to get a more marks. So already I told. So crash program just we can try better our level. In case you may get a high score. If not, don't worry. Better you join a year-long repeated program because we have more time. We are focusing only the need study only. So at that time, during that class time, that the staff will be teaching everything clearly. At that time, you can take a hints. You can clarify your doubts with the concerned faculties. After that, you can in the discussion section, you can discuss it. Oh, why that answer comes. So you can think it and you can discuss with answer with friends or maybe faculties. So lot of, and you can prepare a multiple choice question based on the NCERT syllabus and we can discuss it. So after that discussion, you can get some conclusions. You can get some ideas. Oh, this areas, the questions mostly, if you revise the 10 years question paper means, the mostly they are asking frequently questions from the specific areas, the chapter, the particular chapters. Like this, you can put on analysis. So surely you will get a high score. Surely you will get a, if you following like this means, surely you will get a high scores. Clear. I think the session may be useful. Kindly share your valuable comments in comment box. Thank you, students. We will meet tomorrow. New chapter. Thank you for all.